Hi, today we're going to cover chapter 13 where we're going to talk about meiosis and sexual life cycles. To get us started, let's look just at this picture of this family on this particular side where you've got dad and mom and kids. As you look at the kids, you'll notice that they look a little bit like dad and a little bit like mom, but they don't have all the same things in common. So for instance, like if we look at this daughter right here, she's got light colored hair like mom, but she's got the darker skin and the darker eyes like dad. And that's what we want to learn about in chapter 13 is a little bit about what's going on at the cellular level that causes this to happen. As we go through chapter 13, we're going to go through the following items. First, we'll look at chromosomes. We'll talk some more about definitions. We'll talk about tools that we have to talk about chromosomes, and we'll practice again understanding the difference between haploid and diploid. We'll then talk about the basic sexual life cycles in 3 different organisms. These sexual life cycles are important, especially for those of you that are going into biology too, or maybe going into vet or medical or dental professions. These are something that you'll see over and over again. After that, we'll look at the process of meiosis. The nice thing about the process of meiosis is that it's very similar to mitosis. And so if we've done our work in chapter 12, that makes chapter 13 a lot easier. And then lastly, we'll discuss genetic variation. So concept 13.1 is where we're going to begin. Offspring get genes from parents by inheriting chromosomes. So when I say that I inherited gray hair from my father, I'm not meaning that he actually gave me his gray hair. He's already got gray hair, right? What he gave me was he gave me a gene. Genes are the units of heredity. They are segments of DNA, and this what gets passed to the next generation via gametes. So what my dad did was he gave me a piece of DNA, a chromosome, and how did he give that to me? Through his gamete, through his sperm. So when his sperm and my mom's egg fused together and made me, that's how I got that segment of DNA. Related to genes are things called a locus. A locus is a gene-specific location on chromosomes. Genes always reside in the same location. We number chromosomes from the biggest to the smallest, so we will imagine that this chromosome that I'm drawing right here, it's a really big one, so maybe this is chromosome number 1. If we look at chromosome number 1, it's going to have lots of different information on it. So maybe up here at the top it has some information about hair color, and down here it has some information about shoe size, and down here it has information about eye color. If we look at that specific location where it's telling us about eye color, that is called a locus. And it turns out for every single human being, if you look at chromosome 1 at that specific spot, that's where that gene will reside. Just some terminology that may make things easier for you. When you hear the word gene, think of a generic category. So a gene is going to be something like eye color, right? That's a generic category. Genes come in alternative versions called alleles. Think of the allele as the specific information. So the allele might say something like brown eyes or blue eyes or hazel eyes. So again, a gene, that generic category, always resides in a very specific location called a locus. What we want to do in chapter 13 is we want to begin considering how does sexual reproduction take place? How do we make an egg? How do we make a sperm? So this is a nice compare contrast. Last chapter we were talking about asexual reproduction. So in chapter 12, as we talked about mitosis, remember there were 3 key roles for mitosis. They included asexual reproduction, growth and development, and repair. These require just a single individual. This is where you make an exact copy and there's no fusion of gametes. So what you get is clones. Here are some examples of asexual reproduction down on the bottom. We're no longer talking about asexual reproduction. In chapter 13, what we're doing is we're focusing on sexual reproduction. In sexual reproduction, you have 2 parents. So you've got a mom and a dad, right? Male and a female. And those 2 parents are going to combine and mix their DNA in a way that you get unique offspring, that the offspring don't look exactly like mom or exactly like dad, but they are new, unique combinations. In order to do sexual reproduction, you do need to make gametes. You need to have egg and sperm, and those gametes are made via the process of meiosis, which we're going to learn about. As we continue our talk about sexual life cycles, we want to just continue to make sure that we understand the vocabulary that we're using. Human somatic cells, remember that means body cells or diploid cells, have 23 pairs of chromosomes. When we talk about a human somatic cell, we'll use this type of notation. We'll say it's a somatic cell or a diploid cell, so 2N equals 46. There's 46 total chromosomes, but that's 23 pairs because you've got a number 1 from your mom and a number 1 from your dad, a number 2 from your mom and a number 2 from your dad. So you've got 23 pairs. One way to look at your chromosomes is through this really fun technique called a karyotype. A karyotype is a way that we can take a picture and organize all of your chromosomes. So it's a way to visually look at someone's chromosomes. As you look at the chromosomes, you pair them up according to size. The paired chromosomes are called homologous chromosomes or homologs. This is a really important vocabulary word. We're going to use it a lot. So homologous chromosomes are chromosomes like we've got drawn down here at the bottom that are the same length. So here's this purple one. Here's this green one, right? They're the same length. They're the same shape. Their centromere is in the exact same location. And they have the same genes. So both of these at the top might have the gene for something like freckles. However, they might say slightly different things. For instance, maybe this purple gene says have freckles, and this green one says don't have freckles. In either case, though, these are considered homologous chromosomes. With homologous chromosomes, they are always going to be the same length, the same shape, and code for the same genes. Sometimes homologous chromosomes are drawn as they exist as unduplicated chromosomes. They can also exist though as duplicated chromosomes. So this right here in the black box, that's also a pair of homologous chromosomes. Just to remind ourselves, practice some drawings and make sure we know vocabularies. When we look at a cell and we look in on the nucleus, we've got a mass of DNA and protein all loosely coiled. That's called chromatin. When the cell gets ready to go through cell division, that chromatin is going to condense, and it's going to condense into chromosomes. You can have an unduplicated chromosome, or you can have a duplicated chromosome. Each chromosome will have a narrow region at the center. That region is called the centromere. In an unduplicated chromosome, there are no sister chromatids. However, in a duplicated chromosome, there are 1, 2 sister chromatids. Homologous chromosomes may exist as unduplicated chromosomes. So here's a pair of homologous chromosomes. Those are the same shape and the same length. And this is also a pair of homologous chromosomes. These two chromosomes are the same shape and the same length. They code for the same things. One way to think about diploid cells, when we say that something is diploid, is that diploid cells will always have homologous chromosomes. So if you look at a cell and you can find homologs, or these two chromosomes that are the same length and the same pair, then you know you're looking at a diploid cell. Just remember about how numbering works. In this green box over here, we have 2 unduplicated chromosomes. In this green box, the blue chromosomes inside, we have 2 chromosomes as well, even though they're duplicated chromosomes. Here's a picture of someone taking a karyotype. You can see that they use a microscope. They take a picture of all of the chromosomes in the cell. Then using computer algorithms, what they do is they line these chromosomes up by size and shape. And then you get these pictures that look something like this. You can see that the biggest chromosomes are going to be at the beginning, and the smaller chromosomes are going to be down here at the bottom. In this box right here, what we've got drawn is a pair of homologous chromosomes. So what you're seeing is this. And you can see that the individual chromosomes get successively smaller. And then at the very end, we have the sex chromosomes. The sex chromosomes are called X and Y. Human females are XX, human males are XY. The remaining 22 pairs of chromosomes are called autosomes. Looking at this individual, take a moment and then tell me what the sex is. Hopefully you saw the 2 X chromosomes and you recognize that this is a female. What about this individual? You can see that they have the single X chromosome, and then they also have the Y chromosome. So this is a boy. Again, these pictures are called karyotypes. As we look at the karyotypes, we can tell that this individual is a diploid organism, right? We can tell this is a normal human somatic cell because there are 2 chromosomes for number 4. There are 2 sets of chromosomes, and these 2 chromosomes are called homologs. One of these chromosomes came from the organism's father. One of the other chromosome came from the organism's mother. The 46 chromosomes in a human somatic cell are 2 sets of 23. You got 1 set of 23 chromosomes from your mom, 1 set of 23 chromosomes from your dad. So a diploid cell has 2 sets of chromosomes. For humans, you should know that our diploid number is 2n equals 46. Gametes are going to be special types of cells. So when we talk about sperm if you're a guy or an egg if you're a girl, this is a cell that has a single set of chromosomes. These are going to be haploid cells, and in humans our haploid number is n equals 23. So if I took out one of my eggs and we looked at it right now, each of my eggs would consist of 22 autosomes and a single sex chromosome. In an egg, I'm always going to have an X as my sex chromosome because I'm a girl, right? I've got an X or an X. So the only thing that I can put in my eggs is an X chromosome. When guys make sperm, however, they are going to have 22 autosomes, and then their sex chromosome might either be an X or a Y chromosome. So in that sperm, 50-50 chance. So when you think about families where they're like, oh, I just want a son, or oh, I just want a daughter, really it's that sperm that's choosing whether that baby is going to be a male or a female. On this slide, we see a karyotype, just a nice way to refresh and review. Up here at the top, I can see a diploid karyotype. I know this not just because it says that it's a diploid karyotype, but because I've got homologous chromosomes, right? Whenever I see homologous chromosomes, I know that I'm looking at a diploid cell. On the bottom half, over here on the left-hand side, I know that this is a haploid cell. I can quickly tell that because I do not see homologous chromosomes. I just have one of every type of chromosome. This particular one has an X chromosome in it, so this is either an egg or it might be a sperm as well. And then on the bottom right-hand side, we can see another haploid karyotype that has that Y chromosome. And because it has a Y chromosome, I know that this bottom one has to be coming from a sperm. Again, it's important to remember that the first 22 chromosomes are called autosomes, and then the last chromosome is your sex chromosome. On this slide, we've got a picture of a cell, and what I want you to do is to take a moment and see if you can put some of the stuff that we've been talking about together. So that cell that you can see there right now, what I want you to figure out is how many chromosomes are in that cell. Just take a second and go ahead and write down a number or say something out loud, but actually decide how many chromosomes are in that cell. Hopefully, as you looked at that cell, you realize that there are 1, 2, 3, 4, 5, 6 chromosomes. So this is a cell with 6 chromosomes. Now I want you to look at the cell and I want you to decide, is this cell that's down here haploid or diploid? Is it a 2n equals 6 cell or is it an n equals 6 cell? Again, now that you've had a chance to look at it, hopefully you realize that this is a diploid cell, 2n equals 6. I mean, it does tell us over here in the corner. How could you tell, though, by just looking at the image? When we look at the image, we can see that there are homologous chromosomes. There are 2 chromosomes that are the same shape and length. There are 2 little chromosomes that are the same shape and length. And then we also have these 2 large chromosomes that are the same shape and length. So this cell is a diploid cell with 6 chromosomes on the interior. We're going to practice this in the upcoming slides. I've got 5 or 6 to try through. As we look at each slide, what I'd like you to do is to try to, on your own, figure out how many chromosomes are in the cell, and is it diploid or is the cell haploid? Okay, here we've got a cell. Again, I'd like you to figure out how many chromosomes are inside, and is the cell diploid or haploid? I really do want you to just take a moment, try to write it down. You can pause this if you need more time to think about it. As we look at this cell, let's count up the chromosomes first. There's 1, 2, 3, 4, 5, 6, 7, 8. So I know that this cell has 8 chromosomes. And then I'm going to try to figure out if it's diploid or haploid. As I try to figure that out, I notice that there are homologous chromosomes. There's this little one plus this little one. Those are the same shape and size. Or there's this long and skinny one plus this long and skinny one. And so what this means is that this is a diploid cell. We would say 2n equals 8. Let's try another one. Again, feel free to pause. Otherwise, figure out how many chromosomes and is this diploid or haploid. In this cell, we see that there are 4 chromosomes. And in this particular cell, it is a haploid cell. There is no homologs, right? There's no matcher for this particular cell. Let's try another one. Here you go. How many chromosomes? Is it diploid or is it haploid? All right, in this cell, we've got 1, 2, 3, 4 chromosomes. So we know that there are 4 chromosomes inside. This cell, as we look at it, does not contain homologs. There is no matching chromosome that is tall and skinny. So this cell turns out to be a haploid cell. This cell is best described as n equals 4. Note, haploid and diploid has nothing to do with lines versus X's or duplicated versus unduplicated chromosomes. The way that you can look at a cell and tell if it's haploid or diploid is to look for homologs. So this one often tricks up people in class. Let's keep going and make sure that we understand. Okay, here you go. How many chromosomes, haploid or diploid? Give it a chance. All right, I'm looking at this cell and I see that there are 6 chromosomes. When I look at this cell, that I see that there are homologs. There's 2 that are matchers. Here are 2 that are matchers. And so this turns out to be a diploid cell. 2n equals 6. Here's a really full slide. Let's see if you can do this one. How many chromosomes and is it diploid or haploid? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. All right, I see 10 chromosomes. And it turns out that this is an n equals 10 cell. Why? Because there are no homologs. I cannot find another chromosome that is the same shape and size as the particular one. Here's our last one to try. How many chromosomes and is it haploid or diploid? Try on your own before I give the answer. 1, 2, 3, 4, 5, 6, 7, 8. Okay, so I see 8 chromosomes. And as I look at these, this has homologs because here are 2 chromosomes that are the same shape and the same length. So this is a diploid cell, 2n equals 8. Hopefully this has helped you become a little bit more comfortable with the way that we talk about cells when we look at the karyotypes and we discuss the chromosome number. Now we want to start talking about life cycles. Gametes and egg and sperm fuse through the process of fertilization. When we have a fertilized egg, we call that the zygote, not the baby. The zygote will then grow through the process of mitosis into an adult, and then at sexual maturity, the ovaries and testes in the adult can go through meiosis to produce gametes. This picture hopefully is not a surprise to any of you. This is how the human life cycle works. So again, we'll just go through that image, sort of walking our way through. We start off with 2 haploid cells, sperm and egg. Note that in this picture, everything on the top half of the picture is trying to represent where the organism is haploid. So that's why it's sort of the green color. You can see that there's also those N markings. Egg and sperm are going to come together and fuse through the process of fertilization. As we look at the life cycles, fertilization will always happen at that point. Once we get there, we have created the diploid zygote. Notice down at the bottom, we're now looking at 2N organisms. This is where they are diploid. Okay. Through the process of mitosis, that organism continues to develop into the adult. The adult goes through meiosis. And what does meiosis do? Meiosis is making those haploid cells, and we're right back to where we started. As we look through the 3 basic sexual life cycles, we're going to notice that they differ in their timing of meiosis and fertilization. Mitosis will happen both with diploid cells and haploid cells. Meiosis can only happen in a diploid cell. Meiosis is where you take a diploid cell and break it down into haploid cells. Sexual life cycles are important because they contribute to genetic variation in offspring. And genetic variation, as we think about evolution, is a good thing. We want organisms to look different from one another. As we look at the sexual life cycles, a couple of things to note about the sexual life cycles. All of the sexual life cycles will have a few elements in common. As we look at the sexual life cycles, they're all going to take this basic format. The top half of the image is going to be where the organism is haploid, and the bottom half of the image is going to be where the organism is diploid. There will be 2 processes that every organism goes through. Every organism is going to take haploid cells and fuse them through the process of fertilization. Every organism is going to take a diploid cell and break it apart into haploid cells through the process of meiosis. And so this basic format will stay similar as we look at the 3 different sexual life cycles. Here's the animal life cycle. We've already been through it once, and again, this is your life cycle, so it shouldn't be too much of a shock to you. We have 2 haploid gametes, think egg and sperm, that fuse through the process of fertilization. There they form a zygote. The zygote can grow through the mitosis process to become the adult. The adult then goes through meiosis. In the process of meiosis, it makes haploid gametes, and we're right back to where we started. The next life cycle that we want to talk about is called the alternation of generations. This is what plants use. And as we go through this life cycle, at first it will seem very similar to the animal life cycle, but plants contain some structures that we do not contain in our life cycle. So we'll start the same place as we did for animals. Let's start off with 2 haploid gametes, egg and sperm. So just like us, egg and sperm, these haploid gametes fuse through the process of fertilization to make a diploid zygote. That diploid zygote can then grow up through mitosis to become the big adult. Here's where some of the terminology becomes a little bit different. The big multicellular diploid organism, we call that the sporophyte. We don't call it the adult. This is often the plant, right? So when we look at a tree, we're often looking at the sporophyte. The sporophyte, like a diploid organism will go through the process of meiosis, and it will make haploid cells called spores. These are totally something that we do not have any sort of corollary in our own life cycle. This is unique to what our plants are doing in this particular life cycle. So they create these haploid spores, and these spores begin to go through mitosis. So this single cell that they made that's haploid begins to divide and divide and divide and divide, and we end up with this big multicellular haploid thing that often looks just like a normal plant. This haploid organism is called the gametophyte. It's able to photosynthesize, it looks normal, it's just part of the plant. This gametophyte, as needed, is already haploid, and so what it can do is it can go through mitosis to make the gametes, and we're right back to where we started. This is definitely the most complex of the life cycles that we look at, but you should be comfortable with this alternation of generations. So we have our diploid multicellular stage called the sporophyte, right? That's the big thing down here. The sporophyte goes through meiosis to make spores. You have a haploid multicellular organism, the gametophyte. The gametophyte goes through mitosis to make gametes. Some things that might make this a little bit easier. When you see that ending phyte, think that's the big thing, right? The sporophyte is the big diploid thing. The gametophyte is the big haploid thing. Whatever the sporophyte is, it tells you what kind of things it makes. Sporophytes make spores. Gametophytes make gametes, and that might help you through this cycle. The last cycle that we have, we'll call this the fungal life cycle. In the fungal life cycle, there is no multicellular diploid stage. We're going to start the same place that we started for all the others. You have two haploid gametes, think egg and sperm, that fuse through the process of fertilization and make a zygote. Note that that initial step is always the same in all three of these life cycles. Fungi and some protists are a little bit different though, because this zygote is very transient. It does not get larger. This zygote immediately goes through meiosis and makes haploid cells. Those haploid cells, we are not going to give them any special generic name at this point. These haploid cells can go through mitosis and can become the big multicellular organism that then goes through mitosis to make the gametes, and we're right back to where we started. Again, you should know these three life cycles and feel comfortable with them. That brings us to meiosis. So we're finally ready to talk about meiosis. Meiosis, the entire process is trying to take a diploid cell and create a haploid cell. So we're trying to take something that has two copies of every chromosome, and we're trying to get to a cell that has just a single set of chromosomes. Meiosis is going to be preceded by S phase, so the DNA is still going to have to duplicate before we start. Meiosis is going to take place, though, in two sets of cell division. So we're going to do PPMAT twice. The first division is called meiosis 1. This is when homologous chromosomes are going to pair up and separate. And the second part is meiosis 2, when sister chromatids will separate. Meiosis 1 is when we see most of the unique things happening. There are 3 unique things that happen during meiosis 1 that we're going to point out. Meiosis 2 is going to look exactly like mitosis, and so it'll be very easy to talk about. At the end, when we're done doing meiosis, what happens is the following. We will take one diploid cell and we will end up now with 4 haploid daughter cells. There are 3 events that are unique to meiosis. The first one that occurs during prophase 1 when the DNA is condensing. So just like in mitosis, the DNA is going to be condensing during prophase. So here's my interphase cell. The DNA is all in chromatin. During prophase, what happens? That DNA begins to condense and we see our duplicated chromosomes. Synapsis is the following. The homologous chromosomes, those 2 chromosomes that are the same shape and the same length, are going to find one another and they're going to buddy up. And they often lay on top of each other. That's called synapsis, those homologous chromosomes condensing and being associated one with another. DNA is very fragile, and so it often breaks where the DNA is cut. And so what happens is the cell really quickly is like, oh no, we just cut the DNA, and they go to glue the DNA back together, but they accidentally glue the wrong piece onto the wrong part. And so you end up with chromosomes that look something like this, where you've had this crossing over event. So synapsis is the pairing up that we saw, and then crossing over is defined as the exchange of non-sister chromatid DNA, right? The green and the red, they weren't sister chromatids, but they were exchanging their genetic information. Crossing over in synapsis during prophase 1, it needs to be understood. Here we're going to look at it in further detail. Remember, sister chromatids are held together by a molecule called cohesin. In this image, it's the purple little circles. Non-sister chromatids get broken at precisely corresponding positions. It happens down here in this area. There's a structure that forms called the synaptonemal complex. It's those green images that holds the homologs tightly together. That's synapsis. That's them buddying up, right? So the DNA is broken, but synapsis is holding them tightly together. The DNA breaks are repaired. This is called crossing over. You can see it happening right here. When DNA from one non-sister chromatid is combined to a corresponding segment of another. Again, this all happens right at the very beginning of meiosis and is something unique to meiosis. Crossing over occurs during prophase 1 of meiosis. The next two events that happen are also right there at the beginning of meiosis. So during metaphase 1, homologous chromosomes align. So when we get to metaphase 1, everything's going to line up just like they did in metaphase before, but this time it's going to be homologs that are aligning, right? These buddies that are the same shape and the same length, they're pairing up. So we could say that homologous chromosomes are aligning. Another word for that homologous chromosome pair or the homologs is to call it a tetrad. During anaphase 1, things are going to continue to pull apart just like before, but this time it's homologous chromosomes that are separating. So if we think about what will happen in anaphase 1, homologous chromosomes begin to pull apart. All right. If you can remember these three events, that during prophase 1, synapsis and crossing over occur, that during metaphase 1, the homologs align, and that during anaphase 1, the homologs are separating, you're set to go for meiosis. The rest is going to look very, very, very similar to mitosis. Here's a nice little graphic, gives us a chance to practice some stuff. As we look at meiosis, we're starting off with a cell that has 2 chromosomes in it. This cell is a diploid cell because they're the same shape and the same size. This cell is going to go through interphase. During interphase, it'll go through S phase. During S phase, the chromosomes duplicate. And now we've got this cell with duplicated chromosomes. Still note that this is a diploid cell with 2 chromosomes. We then go through meiosis 1, where we have synapsis, the homologs align, and then the homologs separate. Note, when I look at one of these cells, I now have just 1 chromosome, and this is a haploid cell. So in my first round of cell division, I've already gone through from a diploid cell to a haploid cell. The problem at this point is that this chromosome is still duplicated. I've got the X. I need to get to just a single line. So I'm going to go through another round of cell division. This is called meiosis 2. In meiosis 2, we are going to take the sister chromatids and separate those apart. If we look at just one of the cells that we're left with at the end, there is just 1 chromosome in that cell, and it is a haploid cell. So in this process, we've started off with a diploid cell at the top, and by the time we're done, we're ending up with 4 haploid cells. Now that we've set up the basics of meiosis, I'm just going to take you through a couple slides to continue getting more comfortable with this process. Remember that before we do meiosis, it does need to go through S phase of interphase, duplicate the DNA. In meiosis 1, we're going from a diploid cell to a haploid cell. So we start off with a cell that is diploid. You can see there's 6 chromosomes in there, and they are diploid. And then during prophase 1, synapsis and crossing over occur. During metaphase 1, the chromosomes align up as homologs. During anaphase 1, the homologous chromosomes separate. And then during telophase 1, we're going to have the nuclear envelope reform, and then cytokinesis will already occur. The entire process of meiosis 1 is going from a diploid cell ending up with a haploid cell. That's the purpose there. So in prophase 1, this is when chromosomes begin to condense. Synapsis occurs. This is where the homologous chromosomes are loosely pairing up, aligned gene by gene. Crossing over occurs. Those non-sister chromatids exchange DNA segments. That's structure where the DNA crossing over is occurring is a structure that we call the chiasmata. It just means the cross. And we've got those tetrads. After prophase 1, we get metaphase 1. This is where the tetrads are lining up at the metaphase plate. We can see them lining up right here. Microtubules attached to the kinetochores, just like in mitosis. In anaphase 1, the homologous chromosomes begin to separate. This is guided by the spindle apparatus and occurs because the cohesin molecules get cleaved. Then during telophase 1, the nuclei reform, each half is now haploid, and cytokinesis occurs simultaneously. As soon as we are done with meiosis 1, the cell will immediately go into meiosis 2. It doesn't go through another round of S phase. So we've got 2 haploid cells. that we just made through the process of meiosis 1. In meiosis 2, they're going to go through prophase 2, metaphase 2, anaphase 2, telophase 2. It's going to look very similar to mitosis. The entire purpose of this second round of cell division is we want to go from a duplicated chromosome to an unduplicated chromosome. So we want to go from an X to a line. So in prophase 2, the DNA condenses and the spindle apparatus forms. In metaphase 2, the chromosomes line up. In anaphase 2, the sister chromatids are separated because the cohesion molecules are cleaved. Then during telophase 2 and cytokinesis, the nuclei are reformed, the chromosomes begin decondensing back into chromatin form, cytokinesis happens, and we now end up with 4 unique haploid daughter cells. Now that we're done with meiosis, we can compare what we did in Chapter 12 with Chapter 13. Your textbook has a really good image that goes through these comparisons as well. In mitosis, Chapter 12, it was all about keeping chromosome number the same. Meiosis, we want to change the number of chromosomes. We're trying to go from a diploid to a haploid cell. In mitosis, Chapter 12 was all about keeping everything exactly the same. So the cells that we ended up with looked exactly like the parent cell. Meiosis wants to get differences. Meiosis is going to produce cells that are different from one another and from the cell that we originally started off with. In mitosis, we end up with 2 diploid cells. In meiosis, today, we ended up with 4 haploid cells. This is a great image from your textbook that takes the same cell and it takes it through both mitosis and meiosis so we can compare them side by side. Let's start right here in the middle. We've got a normal diploid somatic body cell where 2n equals 6. Normally the DNA would be in chromatin form, but for simplicity's sake, we're drawing it as though it's already condensed. It goes through S phase of interphase, and now the DNA is duplicated. So during prophase, that DNA would condense. During metaphase, those chromosomes line up. During anaphase, the sister chromatids pull apart. And we end up with 2 daughter cells. These daughter cells look exactly alike one another, and they look exactly like what we started off with. So that's mitosis. Now let's look over here and see what's going on with meiosis. So we're starting off with the same 2n equals 6 cell. We go through S phase of interphase, and that DNA is duplicated. And now we hit prophase 1. The DNA is condensing, but note, this cell looks a little bit different than what we saw over on the mitosis side. Why? Because synapsis and crossing over is occurring right there at the very beginning during prophase 1. Then during metaphase 1, we see that those homologs are aligning. Again, note how that looks very different than what we saw previously. Right? It's homologs aligning, not just a straight line of chromosomes. During anaphase 1, those homologous chromosomes are going to pull apart. And after we're done looking at anaphase 1, we're left with 2 cells. Each of these cells is now a haploid cell, in this case, n equals 3. We don't stop at this process though. The cell is going to go through another round of cell division. That second round of cell division will cause the sister chromatids to be separated. And now we will end up with 4 haploid cells. Because of crossing over, you'll notice that these 4 cells all look a little bit different from one another. Right? And that's part of what's going on during meiosis, is that we're starting off with 1 diploid cell and we're ending up with 4 unique haploid cells. Here's a nice little concept check that I just want you to, as we go through, we'll start at the top and go down. Try to decide if you would answer mitosis, meiosis, or both. Again, it's better if you actually try to write this down. Don't just listen to me and be like, oh yeah, I would have said that answer. So, mitosis, meiosis, or both. Which is required for growth in multicellular organisms? Hopefully you said mitosis. That's one of our 3 key roles of mitosis. All right, the next one. Produces haploid gametes. Is that mitosis, meiosis, or both? Meiosis, hopefully you got that one. All right, third one. Replicates nuclear DNA. Mitosis, meiosis, or both? both of them. Both go through S phase of interphase prior to starting asexual reproduction. Mitosis, meiosis, or both? The answer here is mitosis. All right, next one, sexual reproduction. Is that mitosis, meiosis, or both? It's meiosis. Then our last one, replacing damaged cells. That's mitosis. Really, this slide was a way to review sort of 3 concepts with you. First thing that I wanted to review is that both mitosis and meiosis go through S phase of interphase when the DNA is replicated. Second thing that I wanted to review. From chapter 12, we learned that there are 3 key roles of mitosis. Those 3 key roles are growth, asexual reproduction, and repair. So again, it was just a way to sort of emphasize those 3 key roles of cell division: growth, asexual reproduction, and replacing damaged cells. And then lastly, to think about meiosis, which we just talked about. It's a way of doing sexual reproduction to produce haploid gametes. Ooh, here's another blast from the past. Let's see if you can do it. How many chromosomes are in the cell? And is this haploid or is this diploid? All right, I'm looking at the cell and I see 1, 2, 3, 4, 5, 6, 7, 8 chromosomes. All right, diploid or haploid? Hopefully you recognize that it is diploid. There are homologs in there. Again, if you see the homologous chromosomes, you know that you're looking at a diploid cell. Our last concept is that to talk about genetic variation. Genetic variation produced in sexual life cycles really is important for evolution to work on. Mutations are the original source of genetic diversity. So tens of thousands of hundreds of thousand years ago, there was one hair color for all humans. But then there was a mutation and now there's black hair and there's brown hair. Right? So that's the original source of genetic diversity. Mutations create different versions of genes called alleles. Remember, gene is the generic category, something like hair color, and then the alleles are something like brown or blonde or red hair. The reason, though, that you have brown hair or black hair or blonde hair is not probably because of a mutation. Usually the reason that we have genetic variation or that we look different is because of sexual reproduction. There are 3 mechanisms that contribute to genetic variation in sexual life cycles. Again, genetic variation is the idea that we're all humans, but we all look a little bit different from one another. And that's important evolutionarily, that we all look a little bit different from one another, because that means that maybe if there's a disease that kills Professor Miller, you're a different combination of things, and maybe it won't wipe you out. So there's 3 mechanisms that give us this genetic variation. The first one is called independent assortment. The second one should sound familiar. It's called crossing over. And the third one is called random fertilization. We'll go through each of these 3 mechanisms. You should be comfortable with understanding the basis behind each 3. And again, to remember that these are ways that we get genetic variation. So independent assortment. Homologous chromosomes orient randomly at metaphase 1 of meiosis. So we said during metaphase 1 of meiosis, the chromosomes had to align as buddies, right? What this is saying is that there's no rule that says all of the black ones have to line up on one side and all of the blue ones have to line up on the other side. What independent assortment says is the opposite can happen as well, right? We can also line up like this. Each pair of chromosomes is going to sort maternal and paternal homologs into daughter cells independent of other pairs. Here's an image of this from the textbook. Possibility 1. We can line up the chromosomes with all the blues on one side, all the reds on the other side, or the other possibility. And as we follow this through meiosis, you can see that we end up with very different egg and sperm depending on how we line up those chromosomes. So if you line up in possibility 1 and you're a guy, you can make sperm that looks like this blue one or this red one. But if you're that same guy and you just line up those chromosomes a little bit differently, you can make a 3rd and a 4th different type of sperm. Turns out that this is just a statistics problem, a probability problem. The possible number of independent assortment combination is 2 to the nth power, where n is your haploid number. So for humans, n equals 23. So we can do the math. 2 to the 23rd equals more than 8 million possible combinations of chromosomes. Or in other words, if you just take Professor Miller's ovaries and you're trying to make eggs, just by lining up my chromosomes in different orientations, I can make over 8 million different kinds of eggs. This is one of the reasons that none of my kids look exactly like one another, right? Because I've got 8 million different kinds of eggs. This is because of independent assortment. Another factor mechanism that contributes to genetic variation is crossing over. Crossing over is when homologous portions of two non-sister chromatids trade places, and this will combine DNA from two parents into a single chromosome. Remember, this is occurring during prophase 1 of meiosis. If we think back to the family that we saw at the very beginning of this chapter, we had Dad, and he had curly hair and dark eyes, so he had that chromosome that we can see, whereas Mom had straight hair and light eyes. And then their beautiful daughter here had a nice combination of both Mom and Dad. This could have occurred through the process of crossing over, right? So you can see that she got part of that straight hair chromosome from Mom, but then she got part of that dark-eyed chromosome from Dad. So she's this new combination that's not completely Dad or completely Mom, but she's this new combination. This process of crossing over, we've kept it very simplified. The actual process of crossing over is much more complicated. Instead of just crossing over at a single point, chromosomes usually have crossing over events that occur at multiple locations. So this is an astronomical way of increasing genetic variation. Step 3, random fertilization. It turns out that any sperm can fuse with any egg or ovum. That's an unfertilized egg. So let's leave out crossing over just for a moment and think back to independent assortment. We said that just because of independent assortment, I can make over 8 million different kinds of eggs. Well, it turns out my husband can also make over 8 million different kinds of sperm. When we procreate, I do not tell my husband, all right, I've got egg number 2,433 lined up. Make sure you get sperm number 2,433 lined up, right? Any of my 8 million eggs may fuse with any of his 8 million sperm, which means that just thinking about independent assortment and random fertilization, we can make over 70 trillion unique babies. That doesn't even pull crossing over into there. Adding crossing over adds even more variation. What this means is you don't have a doppelganger somewhere out there in the universe. You are uniquely you. Each zygote has a unique genetic identity. The only exception to this, and it's a wishy-washy exception, is if you're an identical twin. Identical twins are the same egg that then accidentally splits into half, so they do start off with the same DNA. However, even identical twins are going to have changes to their DNA where they're not going to be exactly the same, and we'll talk about that a little bit more when we get into our genetics unit. So just as a conclusion, we want to remind ourselves of the importance of having genetic variation within a population. Natural selection, that mechanism of evolution, needs genetic variation. Sexual reproduction is really important because it helps us have continual genetic variation in a population, right? All of this mishing and mashing of the DNA and changing things up means that our offspring are going to look slightly different than us. So what occurs is that animals that reproduce asexually are actually quite rare. This is an animal over here called a rotifer. They're these small microscopic animals, but they are animals. This is their mouth. They have an intestine, a gut. They have little feet down here at the bottom. These little rotifers reproduce asexually, but they're super weird. And so it shouldn't work, but it does work for them. There is a Science Friday video that you should watch about these rotifers to learn more about them. Just as a reminder, we do have Science Friday videos for Unit 3. You should have watched the following 4 videos. If you haven't, these video links are in each of the weekly modules. You can find them. Chapters 9 and 10 are the fermentation and space plants. Naked mole rats you can find for Chapter 12 and 18.5. And then celibate rotifers you can find in the same place that you found this particular lecture. Keep studying. Our exam will cover those chapters. Exam 3 will cover chapters 9, 10, 12, 18.5, and 13, along with comprehensive questions as always. If you have questions about material or what we've covered, please ask me. I hope that you're doing well in the course and that you're understanding the material. Please reach out if you have any questions, and I'll see you next time.